Thursday, the 1st of April. It's just gone 11 UK time, so it's time to welcome you all to today's Marine Biology Live. I'm going to be joined today by Dr. Ollie Tills from the University of Plymouth, who's going to be talking to us about aquatic embryos in a warming world. Just ask Ollie if you're there, if you can just turn your camera on, say hello.
Hi Jack, hi everyone.
Hey, Ollie. You're right.
Yeah.
So yeah, Ollie's a UKRI FLF research fellow at the University of Plymouth. He's a marine biologist and has training in developmental ecophysiology. He studies the early development of aquatic organisms within the context of their environment. He's developed new approaches to studying the dynamic process of early development using technologies, and he's also led the development of Embryo Phenomics, which is an automated technological platform for the high throughput screening of aquatic embryos. And I think he's going to explain to us what all of that means during his talk. So yeah, welcome, Ollie.
Yeah, nice to be here.
Hi there. Before we start your presentation, I was just wondering if you could tell us a little bit about your journey from being a marine biologist into studying embryos, and particularly the complex sounding technologies that you've been using to study them.
Yeah, so I mean, I guess I've always been fascinated by sort of fundamental questions in biology, so I studied marine biology here at the University of Plymouth, obviously here in Plymouth, and I've been in Plymouth, and I've done a master's in Plymouth and a PhD in Plymouth, so I've been here a long time, and that's put me in a really fortunate position where I've been able to sort of develop a capability that's allowing us to do new types of research centered around early development, and hopefully through the talk I'll kind of give you a feel for why I think that's interesting and sort of important work.
Yeah, it's really interesting when these kind of disciplines kind of come together, and well, I mean that's how you get these kind of amazing findings that you've hopefully going to share with us later on. So brilliant. So yeah, when you're ready to go, Ollie, if you want to turn on your share your screen, I'll disappear.
Yeah.
Let you get on with it. Okay, thanks very much.
Great.
That's all looking good. Yeah.
Cool. Okay, so yeah, sort of a really broad title. So aquatic embryos in a warming world, and I'm going to give you a very brief sort of whistle stop tour of some of the research that I'm doing. So I'm a UKRI research fellow based at the University of Plymouth, and I'm also really fortunate to be supported by Plymouth Science Park with sort of innovation space, so we sort of make new technologies and new approaches. So yeah, so lucky to be here in Plymouth, really. Oh. Okay, so the presence of an elephant on my first slide is not because it's April Fool's Day, so stick with me. If I ask you which of these animals is an elephant, you wouldn't really have to think twice. So I have a 1-year-old daughter, and she knows what an elephant looks like. Down in the bottom left-hand corner, we've got a coral. So this is actually an elephant's ear coral, which is entirely coincidental, believe it or not. It's a more challenging ID, but many people would still be able to make it. You know, it's characteristically coral-looking. So how about the next example? So jellyfish. Which of these images is a jellyfish? Again, I would assume that the majority would choose the top image because it looks like a characteristic jellyfish medusa. This is actually Aurelia aurita, the moon jellyfish, which is sort of common all around the UK and beyond. But it may surprise many of you to realize that actually the other two images on this slide are also jellyfish, but they're just jellyfish at different stages of their development. So in the bottom left-hand corner, we have jellyfish polyps. Get the video playing. So jellyfish polyps, which are sort of very alien-looking animals, effectively blobs of jelly that would be attached to the sea floor, and small parts of that jelly then break away in a process called strobilation. And then those parts of jelly effectively metamorphose to become what we have down in the right-hand corner, which are ephyra, the bottom right-hand corner. So the ephyra has kind of got that characteristic jellyfish pulsation or pulsating behavior, But there are some key differences, and I guess one of them is actually they're a lot denser. You can kind of tell by looking at them, they're a lot more dense than the adult jellyfish. And what that means is that they're having to continually swim to sort of stay in position in the water column. So if they stop swimming, they'll sink. And working with jellyfish in the laboratory, what you find is that as soon as they touch something, they tend to stick to it. So there's a real sort of impetus in these Ephyra stage jellyfish to sort of stay active in the water column in order to avoid that happening. So I find this fascinating, the fact that you've got 3 different developmental stages, 3 very different stages of life in a single species. And as I sort of touched on at the beginning, you know, we have an idea of what an animal looks like, an elephant or a jellyfish, but it's important to recognize that that is really just a sort of snapshot of its whole life cycle. So I'm as Jack said, a developmental ecophysiologist, which is a sort of fancy way of saying that I study how animals put themselves together within the context of their environment. So while making a living, while surviving out in the environment. So that's what really sort of drives my research and my interests. So I've worked a lot with this species. This is a snail called Radix balthica. It's actually a freshwater snail, but we find it in estuaries all around the UK, and we also find it in the Baltic Sea. So it's fairly tolerant of salt. And it's, you know, it looks like a snail, but its embryonic development is 2 to 3 weeks long. And during that time, you know, we've got very different looking forms. So they develop within an egg. So the egg is about 1 millimeter in length. So here you can see sort of 5 embryos all developing within their egg, all doing very different things. During the process, they're changing on a sort of minute by minute and hour by hour basis. You know, so they're moving, they're working. So 4 out of the 5 of these animals, so the ones around the outside, have got a functional heartbeat. And you might be able to make it out if you can see the cursor down in the bottom left-hand corner. You can probably make out this flickering. So that's a heart beating. Many would be surprised to know a snail has a functioning heart. Again, you can see the heart beating here. And in the very early stage, down in the bottom right-hand corner, the heart's only just beginning to sort of flicker. So this process takes about 2 weeks. So it's a 2-week process. And during that time, they're changing in how they appear. So they're changing sort of spatially. They're changing functionally. So they're changing how they work. You know, you're going from, in the middle, we've got a sort of effectively a ball of cells with cilia or small hairs around the outside. And they're using these small hairs to spin around inside the egg. Towards the end of development, if they're so large that they struggle to move around in the egg, but then in the intermediate period, they're sort of crawling around inside the egg. So you can see in the top right-hand corner, you've got this sort of snail gliding and crawling around inside its egg. And some of you might be able to make out the eye spots, so these sort of dark spots on the head. You've got the shell at the back. So you can see the eye spots. Again, they're forming. So the very early stages don't yet have those. So this isn't a passive process. At every stage of development, the animal is exposed to the environment and everything that the environment brings with it. I just wanted to say, actually, I forgot to mention that these animals also, in terms of function, they also have to eat their way out of the egg. So snails have a structure called a radula, which is effectively like a sort of rasping file, which it would use ordinarily for sort of grazing biofilms on rocks, for example. But they actually have to use that radula for eating their way out of the egg. So there, the development of function is crucially important, because if they don't develop it, they then can't hatch. So yeah, so they're exposed at every stage of their development to the environment and everything that the environment brings with it. And this means that human-induced environmental change is directly impacting these animals and the development of these animals. And so it's the nature of these impacts that I'm especially interested in. So Radix balthica, this particular species, they lay eggs within egg masses. So you can see here on the left, we've got an egg mass, which is a couple of centimeters long. You might be able to make out the individual eggs within the egg mass, even on these sort of the images on the left-hand side. So we zoom in here a bit closer. So there's about 100 eggs within each egg mass, which is a few centimeters long. And the animal, the embryos stay within this egg mass for sort of 3-4 weeks, depending on various factors out in the environment. And on the right-hand side, you can see this process in time lapse. So you can see the animals developing, moving around inside their eggs, and them developing in quite sort of close proximity to each other. So if you're lucky enough to be able to visit a shore or a pond or a river, give some thought to this hidden world, because it's going on around you. You might not be able to spot these early life stages, but they are developing, and they're developing out in the environment. So seeing as these lectures were organised by, or are organised by the MBA, I wanted to highlight some amazing work that was done a long time ago by Walter Garstang in the early 1900s. So he was one of the first to really look at the form of the development of form and function in marine larvae. So here we're talking about sort of the free swimming larvae, so sort of the plankton, the early life stages in the plankton. And his work has been really important generally, but it's also impacted a lot of the questions and the ideas that I have. So a lot of the research that I have. And one of the things about Garstang is that he had real fun with it. So he used poems as a way of describing biology, which I think is brilliant. And what the Ballad of the Villager is one of the sort of the most commonly or famous, I guess, if you're in sort of scientific circles, particularly developmental. And so in this video at the bottom, we're looking at a snail veliger. So this is actually a Littorina littorea species, again, we find on UK shores, species of snail. So we can see the velum around the outside, these sort of U-shaped structures with the hairs kind of protruding off it, and sort of, you can see that the hair's kind of moving around, almost kind of looks a bit like an angel. And we're looking down from above, so the shell, so we're looking down from above the shell. And you can see that the hairs are sort of, you know, starting to move and then stopping and withdrawing and then coming back out again. So Garstang described this in the first verse of the Ballad of the Villager. So he wrote, "The Villager's a lively tar, the liveliest afloat. A whirling wheel on either side propels his little boat. But when the danger signal warns his bustling submarine, he stops the engine, shuts the port, and drops below unseen." So what he's done there is effectively describe what we're watching in this video, but you know, over 100 years ago. So it's the idea that as the snail is disturbed in the plankton, or there's some kind of disturbance, it's pulling the cilia and the velum. So the velum is this sort of the structure it's using for swimming, but also for feeding, with the cilia on. It's pulling this in a sort of protective way, in the same way that in a garden, if you tap the shell of a snail, it withdraws itself, the shell down over its foot. So there's lots more to the Ballad of the Villager, but I'm not going to inflict me attempting to do it justice on any of you, but you can find it on Wikipedia. So for those interested, you can always find it on Wikipedia. So hopefully I've set the scene for early development, what it is, some of what it looks like, how it works. And I've touched on the idea that the process is happening within the context of the environment, you know, so within the context of the environment and everything that's happening in the environment. So these animals are sensitive to environmental change. But what I haven't said much about is how we study it. You know, amazing work done, you know, over 100 years ago, but where are we now? What approaches are we taking now to trying to understand what our impact is on early life stages? So very early in my career, I was given some advice which I really valued, which was that I needed to find a better way to be able to do the type of science that I wanted to do. And this came from a PhD student who'd spent 3 years staring down a microscope at embryos developing. And the problem is that embryos don't pause for periods of time that you want to eat your lunch or go to sleep. You know, the process of development is continuous. So after searching and failing to find a solution for being able to do this, we decided to build something ourselves. And the advantage is that we're now able to record the entire development of hundreds of embryos simultaneously, but we're also able to control their environment. So we're able to kind of change the environment and kind of mimic conditions that they might encounter in the future. And because development's continuous, we don't end up with sort of periods of time where, you know, you're not able to sleep or eat. And so there are real advantages, both in terms of the science that we can do, but also in terms of us being able to enjoy that. But we produced vast quantities of video. I mean, essentially the video is the system is a robotic microscope stage with control of the environment on top. And we generate lots and lots of video of developing embryos. And so what we're also doing is developing the software that will allow us to automatically measure everything that we want to in the video. So everything from sort of the development of a heart to the growth of an embryo through to sort of changes in behavior. The aim is to be able to extract as much biologically relevant information as possible. And that's very broad, you know, so we're interested in temporal change, you know, so how things are changing during time, how function is changing, so how the embryos are changing, how they work, but also how they're growing, you know, so how they're growing new aspects of their body. And it allows us to do experiments like this. So this is, again, a snail, Littorina obtusata. So it's a species we find locally, actually all around the UK on shores, so an intertidal species. One of the fascinating things about it as an embryo is that it's got two hearts. So here we've got the larval heart beating in the center, and then we've got the adult heart beating towards the back of the animal. So it's got two hearts, and those two hearts beat with entirely different rhythms, which I find very interesting. And not only do they beat with different rhythms, but they respond to environmental stress in different ways. So a really interesting sort of adaptation to being an embryo. And over on the left-hand side, we can see that the sensitivity of different life stages of this animal are different. So at the very top, we've got an early embryo. Letter B is a sort of mid-stage embryo, and then C is an adult. And these lines show the effectively how an embryo or how a group of embryos is able to survive in a particular environment. So we're going from 100% survival at the start of an experiment down to 0%, you know, at the end of the experiment. So the lower temperature here 36 degrees. Sorry, it's doorbell ringing, which was always bound to happen. The lower temperature means that the embryos can survive for longer, whereas the higher temperature means that the embryos die more rapidly. And what you can see here is that the shape of those lines is very different for the different developmental stages. And actually the embryos seem to be more sensitive, particularly to the higher temperatures. So returning back to Radix baltica, again, looking at effects of temperature, here we've got sort of a fast forward and a playing in real time, but you can see that the developmental rate is very different. So a 5-degree difference in temperature means the animal is developing about 1.5 times faster than the sort of the base temperature. So here a comparison of 20 and 25 degrees. And these temperatures are not uncommon to what the animal would be experiencing. So you can see the impact of a sort of an elevated temperature on its biology. Okay, so this particular experiment tested the response of over 150 embryos. So we had 150 embryos, and we took those and we put them into 3 temperatures. And then we were using, again, this software for making all of the measurements for us. So we were measuring their size, their shape, and their heart rate. And the reason why we use software is because we're talking here about like 30 million images, and we have amazing students at the University of Plymouth, but there are limits to sort of their goodwill and in the enjoyment that they can get from being involved in research. So the computer has the advantage; it can do more with the images we give it, so it can measure more of the animal's biology, more of the embryo's biology, but we can also do it to kind of a whole different scale to if you were making these measurements manually. So if I hit play on this one, so you can see the video. So here we've got the embryos 20, 25, and 30. So this is just a characteristic embryo from each temperature. And then we can see their size, their movement, and their heart rate at the bottom. And what you can see is that the trajectory taken by these embryos during their development is very different in the different temperatures. Yeah, so there's some commonality, you know, they're all increasing in size and they're also showing some changes in movement and heart rate, but there are also some kind of key differences. So their size, their behavior, and their heart rate, so three very different measures of their biology are responding in different ways in different temperatures. So I guess the take-home message from this experiment is that an increase in temperature results in the embryo putting itself together in a different way. And this is both fascinating but also troubling, given that we are increasingly warming the environment that these embryos are developing in. So looking forward, how will these embryos be developing in 20, 30, 100 years time? These are the questions that I'm really focused on trying to answer. And it's more important than ever, I think, to understand the response of biodiversity to what we are doing to the environment. And by biodiversity, I'm including early life stages, because they're a fundamental part of biodiversity, even though characteristically we think of a jellyfish as being a jellyfish, an adult jellyfish, and a snail as being an adult snail. So just to round up, I just wanted to touch on sort of one of the new approaches that we're developing, which is something called energy proxy traits. So I talked about the idea that we're measuring things like heart rate and growth and movement, but we're also developing sort of slightly more, I think bizarre is probably a good description, bizarre ways of measuring biology. So here we're looking at pixel values. So ordinarily you see a video or you see an image, and the smallest component in that image will be a pixel. So effectively a single point in the image, which will have a value. So from say 0 to 255, and 255 would be white, 0 would be black, and you'll have sort of a spread somewhere in the middle. Now the thing with a video is that you can only really see the pixels if it's pixelated, so if the resolution is very, very poor. But actually we can use those pixels and we can use those fluctuations in pixel values to be able to tell us something about the animal's biology. So each of those pixels is represented by a number. And what we're doing is developing approaches that draw on this most basic aspect of an image or of a video, and investigating how these are changing during a video. So how these values change during a video with different frequencies. So that's quite difficult, I think, to get your head around. What we've got here on this plot is the egg around the outside. So you can just about make out this sort of ring of peaks. So that's the egg. So here, the higher the landscape, so the higher the peak of the landscape, the brighter that point on the image would be. And then we've got this sort of a sort of elevated section in the middle, and this is where the embryo sits. So if I play this video, you can see the sort of landscape is moving around, and most of that is down to the embryo and the movements of the embryo. And the flat peak in the middle, like I said, is the embryo. But what's interesting is that if we look at this peak right in the center, so the tallest part of the image, that actually corresponds to the animal's heart. So that's the animal's heart, and the movements up and down correspond to the animal's heart beating. So these changes in the surface of this plot is showing us the animal's biology in a completely new way. And what we can then do with those responses is put them within a spectrum. So effectively looking at how they change across different frequencies. And they're proven to be really characteristic of an animal's biology. So a completely new way of investigating the biology, but something that seems to be a little bit more powerful in many instances than choosing particular types of response or particular types of trait. So I'm going to move on quickly from there because I'm conscious of time. So that was a really brief overview of some of the work we're doing on aquatic embryos in a warming world at the University of Plymouth. So we are in a period of unprecedented technological innovation, and that's really exciting. But I think the challenge really is applying that technological innovation to understanding the biology and our impacts on biology. And that's kind of what gets me out of bed in the morning and what gets me excited about the type of research that we're doing. And I say we because science is very, or at least I think good science is very much a collaborative effort. So I'm really fortunate to work with a great team, and a lot of what I've described is done by them rather than me. So that's important. And actually, during the last year, which has obviously been different, we've been increasingly turning to the public. So our team has kind of grown from, you know, a relatively small group to we've now got over 2,000, I think, volunteers, or sorry, over 3,000 volunteers who have collected over 80,000 pieces of data. And those volunteers are members of the public. So we've been using citizen science via the Zooniverse platform, which we've got a link for down here on the left. And so that's been yeah, really revolutionary in terms of the level that we've been able to take the type of science that we're doing. Okay, so I think that's everything I've got to say. So thanks for listening. I'm back to Joe.
Okay, thanks, Ollie. That was fantastic. Okay, so now there's an opportunity for anybody who's watching to ask questions. So, like I said before, if you wanted to ask questions using the Q&A, which is you should find hopefully either on the top or bottom of your Zoom screen, then you can do that. If you're watching through YouTube, I realise there's a slight delay, but we'll try and pick up any questions or any comments from there as well before we finish. And if you'd like to ask questions in person, then just put your hand up and we'll be able to activate your microphone. So we've got a question from Rowena to kick things off. She's asked, how did you develop your database for these images?
So I suppose it started very low tech. Yeah, so sort of having a hard drive that we, you know, effectively wrote images to, and then sort of thinking, oh, we've run out of space, what do we do next? Oh, well, let's just buy another hard drive, and sort of kind of going through that process again and again and again. And like I say, we've now got to the stage where we have lots of video, and it's yeah, it's a real challenge to be able to both keep track of that, but also recognize that you can't necessarily keep everything. So we're increasingly turning to like online data repositories, so things like Zenodo. So what that allows us to do is take video and put it online so that other people can look at it, because there's been a real sort of boom in data in biology. So there's a lot more data, but a lot of that data is very molecular. You know, so there's lots of genomic data online. But I guess my thinking here is that, you know, having video is in many instances as powerful as having that type of molecular data. And it's the idea that you can kind of like bring everything together. You know, so if, for example, you're a scientist and you're thinking, right, I really want to work on how an animal is spinning around inside its egg, there's nothing better than actually just being able to see what the development of a group of species look like, to be able to then choose the one that is potentially most relevant to you. So I guess kind of a bit roundabout, but I guess the short answer is we don't have any sort of particularly fancy database, but it's a case of trying to make it as accessible as possible, and also being fairly conservative in keeping things. I absolutely hate deleting video, so I tend to just squirrel it away and hide it.
Is that fed into the Zooniverse project that you were talking about at the end as well? Presumably that's all kind of connected there.
Yeah, I mean, Zooniverse has just been really useful because it's a way of analysing video that we would never have got to otherwise. And I think the ultimate aim with the Zooniverse was, so we're increasingly using methods like artificial intelligence. So it's the idea that you can effectively train a computer to be able to measure things that, you know, we can measure, but we don't actually always do particularly well at. And so with Zooniverse, because we've had so many people analyse effectively the same video, you know, you're sort of throwing lots and lots of manual measurements at a particular video, you kind of get a bit of a consensus, and you can then use that consensus to train the computer. And one of the things we find is that even within our own research group, there'll be differences, you know, so different people will perceive things in different ways. Some of the things we measure are fairly clear cut, but other things are actually quite sort of subjective.
Okay, that's really interesting. We've used the Zoom platform a fair bit. We've got a session in May, actually. We're going to be looking at citizen science, so that might well come up then. I was just thinking myself, with have you had a look at any, so looking at sort of intertidal snail species, have you had a look at any of the kind of breeding species like the Trinosactylus, the rough periwinkle, and have you noticed any kind of differences there and in terms of the development and how they're affected by the environment?
Yeah, so yeah, we've actually got a PhD student who's in his final year at the moment, Chris Duane, and he's working on Littorina saxatilis. So he's doing some comparisons of different life stages, and he's also doing that kind of within a sort of geographical context. So he's working with colleagues, colleagues from across Europe. So it's an interesting one because the embryos develop in the animal, in the adult. So they are brooded, and that potentially means that they're sort of experiencing a not dissimilar environment to the adult. So he's sort of he's doing quite a lot of work comparing the response of that type of reproductive mode with, for example, Littorina obtusata, and sort of various other species. I guess one of the ideas with a brooded species is whether or not the parent is doing anything to, protect the offspring. So, so are they moving around on the shore in a different way to try to avoid those hotspots? Because there's a lot of sort of heterogeneity. So there are a lot of differences on the shore in terms of temperature. And so the animal can actually do quite a lot to mitigate some of those effects in some instances.
Okay, that's really interesting. I wonder if when you look into that, I wonder if there'll be differences between the different sort of species and subspecies of Littorina saxatilis and the sort of very closely related. Because I know there's a lot of debate about the taxonomy of the species, and a lot of so that would be very interesting to look at. Cool. We've had a comment from Yusuf. So he said one of the topics that most fascinates him during his studies at Plymouth was yours and other investigations of heterochrony, heterokaryote. And he was curious about whether you'd investigated how temperature affects the timing of developmental events discussed here, such as heart formation, etc.
It's always nice to hear from ex-students. So Yusuf, I guess so yes, it would be the answer there. So we're doing quite a lot of work looking at developmental events, and I guess one of the things we're trying to do at the moment is, so developmental events have been a tool that have been used by biologists for a long time, because what they allow you to do is to compare species that look very different. So I showed you that, if you increase temperature, things change. So for example, the heart will start beating maybe at a different stage in development. So you think of sort of early development, late development, and effectively you've got these events that can kind of move around on that timeline. But when you're comparing different species, it becomes much more complicated, because sometimes, the heart might start beating and then it might stop for a while in one species, then it will start again. So it's very hard to know if you're comparing like with like. So what we're trying to do with methods like the proxy traits, these energy proxy traits, is to try to integrate those with that kind of historical approach. So we're interested in whether or not developmental events, which are effectively just a tool that we're using as biologists, are they the really important thing, or is it sort of the gearing up of the animal just before an event? So for example, with the heartbeat, is it the sort of the twitches and the sort of more subtle responses, which actually you don't pick up if you're just looking for a heartbeat? So so we're definitely still focused on that. And I mean, developmental events were sort of the central thing in my PhD. So I'm very, I've definitely kind of got a vested interest in trying to sort of see that forward as well.
Thanks, Ollie. One of the things that really kind of stood out for me was that, when you listen to these incredible talks, all this information, and one little fact kind of sticks in your head. But the fact that Littorina obtusata have two hearts when they're in their sort of embryonic phase, presumably those one of those hearts kind of gets absorbed and disappears when they sort of develop fully. But I
mean, they're really cool. I mean, the larval heart gets reabsorbed so it becomes part of the gut. I think one of the and we had a student worked with us for quite a long time, currently in Australia, Tabitha. And she did a lot of work comparing those two hearts and how the two hearts responded. And what she found was that the animals that developed the larval heart earlier and maintained it for longer were able to survive stressful environments much better than the animals that didn't have that. And she was looking at why that might be, and seemed to think that it was linked to potentially when the velum started to work. So the thinking is that maybe they have to develop that extra heart to maintain the circulation around the velum, which is something that they only have for a period of development, and then again that disappears. So it's a sort of very clever solution potentially to that issue of how do you provide enough circulation to something that's only there for a short period of time. So really interesting system.
Yeah. Is that a unique feature to that species, or is it something that's quite common?
No, I mean there are a fair few species that do have these sort of larval hearts. I think Littorina obtusata is it's a nice example to work with because they're sort of laid in egg masses. You know, that we can work with them nicely in the lab. They're also fairly characteristic in that you've got the sort of the different colour forms. Yeah, so done lots of work with colleagues have done lots of work with Littorina obtusata and things like hypoxia, so environmental drivers like hypoxia, which are really closely again linked to warming events.
Okay. That's fascinating. Okay, so I think that's about it from the questions. If anyone else has got any questions, now's the time to raise your hand. If not, then we'll be posting the video on YouTube after this, so you can ask questions and chat there if you want to. Okay, so yeah, thanks very much, Ollie. That was fantastic talk. Very nice to have an Easter theme. Just to let everybody know as well, we've got our next talk is on the 15th of April, and it'll be Dave Conway. He's going to be talking about Plymouth Sound as a biodiversity hotspot, and linking that with his work on zooplankton community. So hopefully you'll be able to join us for that. Okay, so yeah, like I say, thank you very much. And just another quick reminder, if anyone's not already a member of the MBA, please consider joining. Yeah, and you can subscribe to our YouTube channel as well, which I should start mentioning now as we're streaming. Okay, thanks very much, Ollie. It's great to have you back in the MBA, albeit virtually, after a long time away. Yeah, so thanks everyone for listening. Goodbye.